Hi, my name is Maria Valdez. It is May 24, 2020. This is Lab 2 for the video results for Bio 225 Microbiology. So here we have the results of the E. coli. As you can tell, it didn't really grow. It was quite frustrating because I kept checking on it every day. And it just didn't seem like it was growing, so I gave it another day for it to grow. I mean, there's only one speck there and maybe another one, but you can't really see it. I honestly can't pinpoint where I went wrong. Perhaps maybe I didn't, grab much of the culture from the tube. But I mean, there is a colony growing. It's just one from what I see. One, actually probably two. It's like one and then another little one there. I mean, I don't know if to perhaps maybe leave it another day, just at least for my sake, just to show me that it's still growing. So the next question is like, explain whether your experiment was successful and whether you obtained expected results. For me, I don't think it was. I was expecting it to be more colonized, especially since I dipped the loop in a bacteria. It's not like I, just barely dabbed it. I actually, scooped in there and tried to get it. I'm not happy about the results because I was expecting more, but there's only so much you can do because of the limited resources. So the next question is, explain how your experiment results apply to real life. Well, if microculture would have grown, I guess that can show that with the right environment and the correct amount of cultures or bacteria, it would have definitely, grown. Especially since it does exist in the large intestine. Pretty sure there's just not one colony. I'm pretty sure there's, not colony, but bacteria. There's higher amounts of it in the colon. I guess also in facts of like, there's been some issues in the past about, E. coli growing on food, like spinach or lettuce, or any other forms of food, such as I believe there was that incident with Chipotle and their pork meat. So I guess this applies in the sense that with the correct environment and the correct resources for the bacteria to grow, it can exponentially grow. And then lastly, describe any experimental errors that occurred and how these might be avoided in the future. It definitely was the first one. So it was a little nerve-wracking. I did try to read the review it well in order to see what materials I needed and how it is that I was gonna go about it. There was a few mistakes I made, but the funny thing is that I made the mistake with the yeast, but with the first two I was actually pretty, I was pretty careful to make sure that I sterilized it before I dipped the loop and after. And also when I did the culturing too, I try to make sure that that was something that I was careful with. Because with the yeast, before I capped it, after I took it out, not that part. It was when I inserted the culture into the broth. After that was done, I forgot to actually sterilize it before I capped it. So I had to uncap it, sterilize it again. I mean, I don't know if that was something, but for some reason it grew better than the first two, which was kind of, yeah, it was kind of interesting. So here, like this one too didn't really grow, which I'm kind of sad about. I could see the streaks if you put it up in the, towards the light, you can see where I try to make the dilution. But with this one too, there's only one. I mean, I don't know, maybe I didn't grab enough for the culture. But that one, that. And then with this one, you can see it better. There are a lot. So it's kind of interesting how this one did really well versus the other two. So that's my video of the results for lab 2 for the culturing and also for plating. Thank you.